All right, so this online lecture is mostly about chromosome shape, and we're going to spend a lot of time on mitosis, which is the process in which our cells are actually going to replicate, in particular the nucleus, how we split our nucleus in half, and the process that goes along with that. So as we dive into the structure of chromosomes, we've kind of touched on that a little bit with the first online lecture, but I want to talk about it a little bit more in depth. When we look at our chromosome, we know that the chromosome is essentially comprised of this double-stranded RNA molecule. So let me just go ahead and set up the pointer. All right, it's comprised of this single linear double-stranded molecule. So we know our DNA is going to be comprised of a strand, and those strands are going to come together to form a double helix. And that's what this first line is essentially talking about. So when we talk about the DNA paired with the proteins that I'm going to discuss, so DNA plus proteins, that term is called chromatin. So, for instance, this DNA picture with the protein present is called chromatin. The protein in particular that is present in this chromatin, so DNA plus the protein has a specific name, and that name is called a histone. So from the images here, these little circular proteins, we call that histones. So anytime DNA wraps around a protein, the specific name of that protein is called a histone. When we're talking about one single unit of DNA and histones together, or DNA and proteins together, that is called a nucleosome. So in the image here, of course, the red line is your DNA. This pink protein is your histone, but one unit of them together is called a nucleosome. And when our nucleosomes are organized, they're organized in this zigzag shape. So if you see here, you can see it really well. It kind of goes up, down, up, down, up, down. This is going to be called a 30 nanometer fiber shape. I know this is a lot, but just make sure you get some flip cards with this type of stuff. But a 30 nanometer fiber shape. So when you see DNA that's wrapped around histones in this zigzag orientation, that particular shape is 30 nanometer fiber shapes. So when the 30 nanometer fiber shapes get together to start to get closer, they're going to begin to form these loops. So you're going to have the 30 nanometer fiber shapes as it says here, and then you're going to have these pieces of DNA that just loop out and form a loop. That loop is called a radical loop domain. So this is just space between our 30 nanometer fibers and other 30 nanometer fibers. It's going to be a loop called a radical loop domain. Once we look at the level of compaction within the nucleus, we're going to realize that some areas of the nucleus are going to be highly compacted, meaning we've taken all the DNA and squished it together. That's going to be heterochromatin. So the dark spaces are areas in which DNA is really, really tightly wrapped around each other. But areas in which DNA is not tightly wrapped around each other, that is called euchromatin. So in this image here, the white areas are euchromatin, the dark areas are heterochromatin. So putting together everything we've seen so far, of course, our DNA double helix is here in this spiral format. Once we begin to wrap it around the protein in which we call a histone, now that is called a nucleosome. Those nucleosomes form these zigzag patterns called 30 nanometer fibers, and eventually big loops of those 30 nanometer fibers form these radical loop domains. And then eventually they all compress and compile to form a chromosome that we know and see. So this is just a detailed account of how we get to this chromosome. I think in the first lecture we kind of discussed this part and we skipped over everything here in the middle, but this part is really critical as well. All right, so now I'm going to cover the cell cycle. Now the cell cycle is it has a lot of terminology, so we're going to go through it slowly, and I do not mind covering this briefly in class. But make sure if there's questions, you give me some specific questions so that we don't repeat the lecture in class and also had you do the lecture online. So when we talk about the cell cycle, this is just a series of step-by-step events that are going to lead to cell division and, more importantly, nuclear division. So first, we want to again revisit our chromosome. When we study chromosomes and how they look and how many we have, we call that field cytogenetics. So genetics is our genes and the instructions to make proteins, and the cyto is the cell, right? So understanding the genes of the cell forms cytogenetics. When we're talking about karyotypes, a karyotype is going to show all of the chromosomes that are found within a organism, and it's going to tell us stuff about the size, the gender of the organism, as well as the number of chromosomes we have in each particular chromosome position. So this is an example of a karyotype. This is actually a human karyotype because we can see there are 23 pairs of chromosomes. So you see each chromosome is kind of in these sets of similar chromosomes with it, so it's a pair. Although sometimes they have a curve here or there. But we're able to see after the 22, you see this final set here. This final set here is actually what we call a sex chromosome, so it determines the gender of what you are observing. And if you did not know, the XY chromosome is representative of a male, as opposed to XX, which is representative of a female. So this would be the 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 karyotype of a normal male, because there's no chromosome abnormalities here. And we'll talk about some other things later, some things like Down syndrome or Klinefelter syndrome, in which you can look at this and you'll be able to see there's going to be a certain number of additional chromosomes or certain missing chromosomes along the way. All right, so again, what you see here is humans, again, have 23 pairs of chromosomes, which means we have 46. The first 22 pairs of chromosomes we called autosomes. So chromosomes 1 all the way down to 22 are chromosomes that are going to affect just general things that you need to make a human. So these are going to be 22 pairs, but then the final pair is called the sex chromosome. So again, XX is female, XY is male, and this picture is just showing just that one Y chromosome, showing that, we have the Y chromosome present. So whenever they're doing gender tests, they can actually look and see, let's say the mother who is XX, if they look in her bloodstream or if they look at genetics and they see Y chromosomes present, that must mean there's a male child. If they don't see any Y chromosomes present, that can also mean that it's just a female, some of those things. So when we're talking about sets of chromosomes, we usually use these two terms, diploid or haploid. So for a set of chromosomes to be diploid, we also call this 2N. Think of N as a one individual chromosome. So you basically have two chromosomes here. So humans have 23 pairs of chromosomes. So humans are known as being diploid because all of their chromosomes come in a pair, right? Because it's 2N. So they're diploid or 2N, and humans are diploid. However, if you only have one set of each chromosome, so for instance, if you looked at this picture and it wasn't two chromosomes in position 6, it was just 1 in 6, 1 in 7, 1 in 8, 1 in 9, blah, blah, blah. That's going to be haploid or N. So that just means you have one set. If something is haploid, we can also call it a gamete. And gametes only have one member, one component of each pair of the 23 chromosomes. You see gametes in your reproductive cells, such as your sperm cells for our guys and our egg cells for our girls. That's because if you're mating, let's say you're a female and you're mating with a male, you want to give one set of your chromosomes, and they can give one set of their chromosomes to make a complete organism. If you're giving two sets and they're giving two sets, then now you have 4, and 4 is not what we need. We need 2. So only in your sex cells or your gametes, your egg and sperm cells, do you have one member of each pair. Everything else in your body is going to be diploid or have 2. So again, this is just reinforcing that idea. Haploid cells are going to just have one. You see one pink, one purple, one yellow, versus diploid have 2. There's also other versions called triploid and tetraploid, but this is not very common in the animal kingdom as much as it is in other organisms. All right, so when we're talking about diploid or 2N species, If they're part of a pair, so let's say number 4 is these 2 chromosomes and number 4, we call them homologs of each other. So they are known as being homologous chromosomes. So when we're looking at our homologous chromosomes, I'm actually going to jump forward to this picture and then I'll go back, is an allele is a certain space on a chromosome. So for these pair of homologous chromosomes, you can see that there is an allele that says whatever gene this is going to be blue, and the other pair says whatever gene this is going to be purple. So an allele is a certain position on a chromosome. If we're talking about whether you have a widow's peak or not, you're going to get one gene from mom, one gene from dad. Let's say we're at the bottom with this red one. This gene says have a widow's peak. This gene says have a widow's peak. You will have one, as compared to these other ones in which this says have a widow's peak. This one says do not. You're going to have somewhere in between, and we'll talk about that. So again, an allele is the same place on a homolog that carries that gene. So we're basically just comparing and contrasting between chromosome 1 and the pair for chromosome 1. That's what an allele is kind of discussing. And again, highlighting that there. So as we go into mitosis, I want to introduce again the cell cycle. So all of your cells in your body, they start off very small, of course, and then they're going to grow. And as they grow, they're going to either do one of two things. They're either going to stop growing completely, or they're going to divide. It's like a balloon. So if you either blow up a balloon, you can realize if you keep blowing it up, it's going to pop, so you can stop it, or you can pick up another balloon and start blowing that one up. So all cells are going to do that. So the cell cycle is the set of stages. So you see right here, we're going to cover, it's the set of stages that a cell has to go through to be able to go from not dividing to dividing. So going from growing to dividing, and then continuing to grow, and divide. So these are the specific stages that the cell has to go through. So the first stage is called G1. So G1 is called, it's just the first stage. It's known as the gap 1 stage. Let me just clarify, make sure I don't have it. I do have it expanded a little bit later. So I'll quickly go through this part. So G1, again, is just the first gap phase, which I'll tell you about what happens there. S phase is S for synthesis. Synthesis means make, so we're going to make a lot of stuff here. G2 is the second gap phase, and then finally the M phase is mitosis, in which we undergo mitosis and cytokinesis. So when you're looking at the cell cycle, although there are different phases within it, they can be broken down into 2 main phases, which is interphase. So that's the blue line that goes around the this image here. Interphase is comprised of G1, S, and G2. And then the final phase is M phase, which is composed of mitosis and cytokinesis. So most of your cells in your body are in interphase. That means your cell is not dividing, it's just doing its cellular thing. So this is an image of what it looks like. The blue is the genetic material. And then, this is a cartoon picture of it there. So most of your cells in your body are just, doing their cellular activities, nothing too major. And then there's some cells in a special phase called G0, which cannot divide. So an example of that would be your brain cells or your neurons, also your muscle cells. So once you get a certain number of brain cells, you cannot get any more. So what you're born with is what you have. That's why, unfortunately, sometimes people go through different traumatic brain injuries or things like alcoholism or different drugs, and you can't rebuild those neurons. Also, that happens with muscle cells as well. So if a cell does not divide, if it never divides, it's always in a G0 phase. G1, think of the G as growth, because normally what happens in G1 is your cell is just growing, it's going through normal metabolism. It is essentially the stage that happens after your cell just divided. So your cell is essentially just growing and doing normal cellular behavior. The S phase is where DNA replicates. So on our online lecture, we haven't fully covered DNA replication, but when we do, if you ever got asked a question, which stage of cell cycle does DNA replicate, it is the S phase or the synthesis phase. This is also where we make those histine proteins as well. And finally, the G2 phase Instead of growing, we're starting to create more proteins that we need for division. Because remember, if you're talking about your cell is growing, 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 growing in G1. When it gets to S phase, it duplicates its DNA. So your cell can't just stop here because it has two sets of instructions on what to do. So it has to go to the next phase, which is G2, so that it can start making more proteins to eventually prepare for mitosis. If it ends up here, this can cause a lot of diseases or a lot of cellular problems, or the cell might naturally just die off so that it can, it won't be dysfunctional. So the M phase, again, it consists of mitosis and cytokinesis. So mitosis is the nuclear division. So remember, our DNA is going to be in our nucleus, and we have to go through a very particular process of taking our nucleus and dividing it in half. We want to make sure that we are dividing our instructions very carefully so that we don't start making cells that have the wrong instructions and will do the wrong things. That leads to diseases, that can lead to cancer, that can lead to some really bad things. So we want to be very careful with that process, and that whole process is called mitosis. That's where you duplicate your nucleus or divide your nucleus to get two identical nuclei. It has 5 stages. We'll go through each one: prophase, prometaphase, metaphase, anaphase, and telophase. Now, sometimes you do not see prometaphase. It just goes from pro to meta. But in the book, it covers it, so I'm going to cover it for you. And finally, cytokinesis is where you've already split apart your nucleus, so now your cell has 2 nucleus, and now you just split apart the cytoplasm, which is all the cytosol and other organelles inside the cell, so that you have 2 separate cells. All right, so the first stage is prophase. So prophase begins when chromosomes become visible. So the reason that this is important is, if you remember a few pages back, normally your cells, when they're in interphase, they are not in a chromosome structure. They're just loose, and the cells are not tightly compacted. But whenever we switch over to prophase, your chromosomes start to become visible. So it goes from chromatin, which is just more stringy, to chromosomes, which are more organized. And again, for them to become visible, it occurs through this compaction of the chromosomes. Another thing about the chromosomes, if you see here in this little blue dot in the little image here at the bottom, is attached to each centromere, which remember is the middle part of the chromosome, is this, think of it like a handle, is a little handle called a kinetochore. So attached to the middle part of the chromosome is a little handle called a kinetochore, and microtubules, which you may remember from cytoskeleton, is going to be able to attach to that. So eventually we're going to do a lot of pulling, and think of your chromosome having a handle so that the cytoskeleton can grab the chromosome and pull it wherever it needs to go. So the centromere is the middle of the chromosome, the kinetochore is the handle, and the microtubules are the strings that will eventually attach. And then the mitotic spindle occurs, which is essentially another term for the strings that we're going to use to be able to pull apart our chromosomes. The final thing that begins to happen in prophase is our nuclear envelope begins to dissolve. So if you remember, our nucleus is inside of an envelope where nothing gets inside, and it begins to dissolve because we have to reach our chromosomes. Remember, our cytoskeleton and everything is on the outside of our nucleus, so we have to first dissolve the nuclear membrane so that it can reach inside and grab the chromosomes. Again, the mitotic spindle is the string that's going to be able to connect, but you can see it a little bit closer here. The centromere is the middle of the chromosome. The kinetochores are the blue pieces that are the handles, and then your microtubules will attach to each side. So in prometaphase, it's basically extending what prophase is. So in prophase, we talked about how the nuclear envelope starts to break down. In prometaphase, it breaks down completely. We talked about the mitotic spindle forming. In prometaphase, it completely forms. And again, our kinetochores are going to be fully attached to our chromosomes, and our centromere in particular. And then finally, the chromosomes begin to move towards the middle of the plate. Now, they are not in the middle of the cell, but they're starting to move towards the middle of the cell. So if we go back to our other picture, all we see here is that they're starting to condense a little bit more. but as we go deeper into prometaphase, they're starting to align in the middle of the cell. So I believe the way this picture is kind of vertical. All right, so metaphase, meta means middle. So anything is meta typically means middle. So this is where all the cells, chromosomes, align or line up in the middle of the cell. So this big blue area, this big red area is the cell itself, and the blue area is the DNA. It's going to line up in the middle. So each chromatid, each chromosome is completely in a chromosome format, and the main thing that happens in metaphase is it lines up in the middle. Now remember, although it's lined up in the middle, like you can see here in this picture, it still has a kinetochore which is attached to the microtubule or the string. So keep that part in mind. So metaphase is where it lines up in the middle and it begins to be attached to opposite sides of the cell. Anaphase, think of Ana as separate. Ana is where the sister chromatids or the pair, remember it's an X, this is where your X begins to separate. So imagine if you look back here, you have an X. I'm looking at the magenta one at the top. If you look here in this image, they've separated, they've been pulled apart. So this kinetochore on each side, the microtubule on each side pulls it apart so that they go to opposite sides. Remember, our end goal is to make two different cells. So if we start to get some genetic material on the left and some to the right, this is going to be much easier for us to split the cell apart down the line. So in anaphase, they're going to separate and move towards opposite sides. It ends when the chromosomes have reached the pole. So when they have reached their side of the pole is when it's going to end. And finally, telophase is when the chromosome starts to return to anaphase-like tradition. So telo usually means end. So at the telophase part, we're basically done. So we're ending everything that we have. The chromosomes have moved to their sides, and we're going to start to begin to separate the cell. So the difference between anaphase and telophase is telophase, everything's starting to dissolve. You're starting to form, have your chromosomes decondense and start to uncoil and get back into a chromatin format. And then you also start to form a new nuclear envelope. So you see in the picture here, they're starting, although you can't really see the chromosomes there, they're going to start to become more loose like the chromatin format, and they're going to also begin to get the nuclear envelope around them as well. So the final step, telophase and cytokinesis usually go hand in hand. They kind of happen at the same time. But as your nucleus is starting to get a nuclear envelope and get back into interphase type conditions, cytokinesis is what happens where we finally divide the cytoplasm. Remember, everything we did before was the nucleus. Now we're dividing the cytoplasm evenly to make sure that 50% goes into one side, 50% goes into the next side. So like I said, it usually happens at the end of telophase and kind of overlaps the mitosis before we get all the way into interphase. And what happens is, think of like a drawstring bag, right? Think of an open drawstring bag, anything with a drawstring. If you pull both sides, that hole is going to start to get smaller, and it basically will start to form a plane. So you see, imagine if this was a big open area and this was a drawstring, you're going to basically kind of snip off or cut off the cell at this point. And this little dip that forms here is called a cleavage furrow. So just like we know the word cleavage means split, from what we did with cellular respiration, a cleavage furrow is this little split, is this little dip that occurs here. That's going to be where we can separate and deepen the cytoplasm. So, all right. So cytokinesis in plants is much more simple. Instead of having a cleavage furrow, what we see is little vesicles or little bubbles form in the middle of the plant cell. Remember, the plant cell has a cell wall, so it undergoes everything normally as far as anaphase, telophase, metaphase, all of that stuff. But what you see is little bubbles form in the middle, and then the little bubbles turn into medium bubbles when they merge, and then eventually they turn into one large big bubble, and that's going to be your cell plate that forms. So that's how you keep that nice square shape for plants. It's much easier in plants. So again, this is going to be an image depicting what we just covered. So again, our prophase has both prophase here and prometaphase. You can see in prophase, the nuclear envelope starts to dissolve, your chromosomes start to become condensed, you start to see microtubules forming. In prometaphase, nuclear envelope is completely gone. Your chromosomes are completely attached to their microtubules and they're starting to come towards the middle. Metaphase, they're completely in the middle, completely attached. Anaphase, they're separated. In telophase, they begin to get back to interphase type conditions and have a cleavage furrow forming in the middle. And again, this is just another image putting all that together as well. Prophase, prometaphase, metaphase, anaphase, telophase. This is an image from your book, so I wanted to, again, highlight what the book has just because I think it's important for y'all to have a similar analogy. And this is, again, showing all the different stages and all the different areas that were covered so far. All right, so when we talk about our daughter cells, what do we end up with? We end up with a daughter nucleus that has the exact number and the exact kind of chromosomes the parent cells had. So it is the same and identical. There's going to be something we talk about later that's going to be a different number in a different type of chromosome. But anytime we do cellular division under mitosis, it is going to be exactly the same number and exactly the same kind of chromosomes. So this is, again, if you stepped on some glass and you cut your foot, you don't want your foot to start forming cells that are supposed to be in your arm. They should be the same exact cells that were there before. And this is what happens in mitosis. The organelles are also split evenly, so we won't cover that, but they make sure there's an even split. And remember, our mitochondria and our chloroplasts in plants have their own DNA, so they do their own thing. So when we're talking about mitosis, we're not covering mitochondria because they divide on their own way. Also, when we're talking about the cell cycle, there are a few terms here. One is called cyclin or cyclin-dependent kinases. So if you see, this is an enzyme, and these are enzyme proteins that control if and when your cycle moves forward. Now, this is really important. We're not going to go into this in depth, but if you have any type of cell bio class or any upper level biology class, you will. And we won't cover them like we should, but basically what happens here is your body will be able to stop the cell cycle when it needs to stop it or tell it to go when it needs to tell it to go. So for instance, if you're supposed to have 46 chromosomes and during your duplication stage, so at the end of the synthesis stage, S phase, if you realize you have 46 times 2, what is it, 92, something like that. Let's say it's 92, right? If you had 95, then they'll realize that there was something wrong, and this will stop the cycle from moving forward. You don't want to move forward with 95 chromosomes because you're going to have an uneven number of chromosomes. You want to have the right number of chromosomes. So this is going to help regulate if and when the cycle moves forward. So this is called a cyclin or a cyclin-dependent kinase. So this will be a good question as part of one of my bigger questions. What would you do if you realized your cycle, you had 100 chromosomes and you're supposed to have 85? What type of protein will probably stop the cycle from moving forward? It would be a cyclin. And finally, there are other key molecules that can also play a role besides cyclins. One is hormones. So plant hormones can promote certain types of mitosis, as well as steroids. So there's tons of steroids. If you think of, I'm thinking of I can't recall the major one that people usually use, corticosteroids or different steroids you can use for your skin, different topical agents that has been used to help with wound healing and help to decrease scarring in different tissues. So some steroids can stimulate growth and mitosis. Also, there are different type of protein growth factors that can stimulate cell growth. But there are some drugs, especially a lot of our cancer drugs, that can stop the cell cycle at certain parts. So as I mentioned before, one of the tricks that cancer uses is that even though it begins to duplicate with the wrong number of chromosomes or something is wrong in the cycle, it can interfere or bypass those cycling so that it keeps growing. So cancer is typically uncontrollable cell growth, and that's why unfortunately it's associated with tumors and things like that. So a lot of times cancer takes over your cell cycle system and it just grows and grows and grows. But a lot of the drugs we have on the market work by targeting cancer cells and putting a stop to the uncontrollable growth by inserting a cyclin or inserting something that will limit that cell's ability to duplicate. And finally, so we covered how this works in animals and plants and bacteria. They divide by a very simple process called binary fission. So if you look at fission, there's two S's, put a little line in the middle. That's how you know it's separation or you're breaking it apart during this. Only thing that happens is in bacteria, the DNA will duplicate or replicate. Then you begin to form a cell wall that's going to kind of be like that cleavage furrow that just splits it in half and you have two cells. So the only thing that happens in prokaryotes or bacteria is the DNA divides. It begins to constrict or form that drawstring cleavage furrow that we talked about before. And the next thing you know, you have two bacteria. That's why bacteria can replicate really quickly. So something like E. coli replicates in about 20 minutes, which is why if you get food poisoning, you feel sick. You can feel sick later that day because it can duplicate very quickly. Again, this is just showing that in a more complicated way. This is your cell that duplicated. It's going to have a separation. More specifically, it's called septum. Think of like a nose ring. A lot of people get septum piercings, which is that middle part of your nose. It's going to form a separation, and then the cells are going to be able to duplicate. All right, so that's the end of our online lecture. It was a little bit longer than anticipated, but I hope that you found some value in it. And this is going to serve as what you're covering in class for the week that I'll be out. So have a good one.